Imagine a world where the very essence of our cellular health is safeguarded by tiny protective caps. These caps, known as telomeres, function much like the aglets on shoelaces, preventing the ends of chromosomes from fraying and sustaining their integrity. However, with each cell division, these telomeres gradually lose some of their DNA. Eventually, they become so diminished that they can no longer facilitate further cell division, leading to a loss of protection for chromosomes. This decline in the number of cells capable of dividing can significantly impair the renewal processes of tissues and organs, ultimately affecting their ability to function healthily. While telomeres naturally shorten as we age, individuals with telomere biology disorders, TBDs, such as dyskeratosis congenita, experience this process at an accelerated rate. The pivotal role of telomeres in the aging process and age-related diseases has made them a focal point for scientific inquiry. Recent research conducted at Boston Children's Hospital is enhancing our understanding of telomeres and is setting the stage for innovative treatments for TBDs. A revolutionary approach to telomere lengthening. For over 10 years, Dr. Suneet Agarwal, a co-program leader at Boston Children's Hematopoietic Stem Cell Transplant Program, has been on a quest to discover methods to lengthen telomeres and reverse cellular aging. His research has primarily concentrated on telomerase, an enzyme responsible for rebuilding shortened telomeres. The intriguing question that arose was whether we could manipulate telomerase to aid in telomere maintenance, potentially leading to new treatments for TBDs. This inquiry prompted Agarwal and his colleague, Dr. Neha Nagpal, to delve deeper into the subject. Recent advancements in chemical engineering have produced enhanced synthetic RNAs with therapeutic potential. However, certain classes of RNA present engineering challenges due to their size and functional complexity. The telomerase RNA component, TERC, a long non-coding RNA, has been shown to extend telomere length in human stem cells. To tackle the complexities associated with TERC RNA, Nagpal and Agarwal devised an enzymatic method capable of stabilizing RNAs of any size. Their research demonstrated that this engineered TERC, eTERC, could operate within human cells, exhibiting a lasting and targeted effect. Upon introducing eTERC into various cell types, the research team discovered that a single exposure could significantly increase telomere length in human stem cells, with effects lasting approximately 69 days, equivalent to several years in human terms. Remarkably, eTERC did not disrupt normal cellular mechanisms. What's remarkable about this approach is that we can provide a temporary boost to telomeres without interfering with other natural cellular processes, Agarwal explains. It has a specific effect on cells and then dissipates. The next challenge is to find a method to deliver eTERC to cells outside the laboratory. Agarwal believes this will likely involve a combination of strategies, including nanotechnology and small molecule agents. He remains optimistic about the potential for such innovations. At Boston Children's, we are committed to developing and testing every one of these strategies until we find effective treatments for TBDs. He asserts, exploring new therapies for telomere biology disorders. In previous studies, Agarwal and Will Manhertz, an MD-PhD student in his lab, discovered that the addition of thymidine could dramatically lengthen telomeres in cell lines and stem cells derived from patients with dyskeratosis congenita. Building on these promising results, Agarwal is preparing to initiate a Phase 1 clinical trial to assess the safety and tolerability of a combination therapy involving deoxycytidine and thymidine in both pediatric and adult patients with TBDs. This study will also investigate the clinical and biological impacts of this therapy on patients suffering from TBDs. Check's overview of Boston Children's Hospital. Other research teams at Boston Children's are dedicated to unraveling various genetic variants that influence telomere length, maintenance, structure, and function. However, these genetic variants can lead to a wide array of effects regarding the severity of TBD symptoms, the age at which symptoms appear, and the specific organs affected. For instance, some individuals with variants in TBD-related genes may experience severe bone marrow failure during childhood, while others may develop pulmonary fibrosis or liver disease in adulthood. There are also cases where individuals never exhibit any symptoms at all. Individuals with variants in TBD-related genes often seek clarity on whether they will develop severe disease, explains Dr. Vijay Sankaran, a physician scientist at Dana-Farber, Boston Children's Cancer and Blood Disorders Center. However, different families may carry different mutations, and even within the same family, members can experience varying degrees of impact. Sankaran and his team, led by MD PhD student Michael Poshla, hypothesized that these differences might stem from the interaction between TBD-causing genetic variants and common genetic variations associated with telomere length in the general population. Utilizing samples from the extensive UK biobank, they developed polygenic scores to estimate its combined effect and applied these estimates to various patient cohorts. Their findings indicated that both rare high-impact gene mutations and common low-impact genetic variants independently influence the development and severity of TBDs. For example, individuals with severe early-onset TBD often had polygenic scores linked to shorter telomeres. This suggests that numerous common genetic variants, which have a slight effect on telomere length, may collectively influence whether and how severely someone develops a TBD. This could also clarify why relatives with the same rare variation experience different outcomes in TBD development. While it is premature to determine the clinical applicability of these findings, Sankaran is hopeful that they will lead to future research and a deeper understanding of TBDs, as well as other genetic disorders that present similar challenges. Families with rare genetic variants are eager for answers about what to expect, he states. We're finally making progress toward providing some clarity. To learn more about the bone marrow failure and myelodysplastic syndrome (MDS) program, visit the Boston Children's Hospital website.